Experiences like the ones that we just heard about that motivate all of us at PacBio to work hard every day to bring ever more advanced products and applications to all of you so that you can solve more cases, help more families, create more productive crops, and do all the 100 things that you do every day. It's been that motivation from the very beginning of the life of the company. As David said, this year is the 25th anniversary, and having had the fortune to be part of the entire quarter century, it is a great privilege and an honor really to share with you some of the moments of its history and give some perspectives on the impact that it has had. Since this is a reason to celebrate, I do have a prop this time for my presentation, so maybe we can bring that out. Terrific. Thank you very much. So the company was founded in 2000, and it was a private enterprise at that time, so it's actually not that easy to find the original documentation. However, I did find the certificate of amendment to the amended and restated certificate of incorporation of PacBio. You got to admire the lawyers, and there it is certified that the company was founded on July 14th, 2000, under the name Nanofluidics. It was founded by these two individuals, Steve Turner, who of course was longtime CTO of the company. At the time in 2000, he was a graduate student in Professor Harold Craighead's lab in the Applied Physics Department at Cornell University. And the reason why they called the company Nanofluidics is because Steve's PhD project was to study the behavior of DNA molecules in nanometer-sized fluidic structures. So you see that where the name came from. So it's actually not the DNA sequencing that was the first business strategy. 3 years earlier, I had arrived at Cornell University as a graduate student, and a few months later formulated the core idea that is, as you can see, watch DNA polymerase make DNA, thereby sequence. I found a PhD advisor and Professor Watt Webb in the same department and built a lab in the basement of Clark Hall, room G2A, where I tried to pursue that idea. The four of us started collaborating at that time, and to summarize about 7 years of work, at Cornell University, we were able to describe in the science paper, we were lucky enough to get the cover, that the zero waveguide nanostructures, which of course Steve invented, were about 100-fold more powerful microscope to observe single molecules. And we, for the first time, also described that we saw hints of enzymatic activity. So polymerase causing these pulses in black that you see when everything is there in the reaction, the controls in red when you leave something out. And that was the moment where Nanofluidics as a company switched over the business strategy from the DNA in the fluidic channels to the DNA sequencing goal. This data and some other accompanying data then led to the Series A funding. Two investors in the Bay Area gave us some money and strongly recommended that we should come to California. I think in part also because they traveled to Ithaca, New York in January to celebrate the closing of the fund, and it was clear that they never ever wanted to do that again. We occupied about a fifth of a building in Menlo Park Business Park, which you can see is still the same and actually a stone throw away from the current headquarters. So you see PacBio headquarters at present is in the bottom left there. This was the first team that occupied that space. The dog's name was Darwin. And as you can see from his body mass index, he was very clever running around from cubicle to cubicle all day getting treats from everyone while we were working there. A year later, we renamed the company from Nanofluidics to Pacific Biosciences, or PacBio. The obvious reason was that we weren't doing anything in nanofluidic structures, but I, for one, had kind of grown tired of trying to spell nanofluidics on the phone to people, and by the time I got to the FLUI part, I wasn't even sure. The reason that you can't tell whether any of these are spelled correctly or incorrectly is probably a good reason also to rename the company, and this is the first picture that I know of after we had put on the new logo and the new name of the company. And then in 2009, we were finally able to demonstrate that indeed it is possible to sequence DNA by observing single DNA polymerase molecules in 4 colors, and in this what we call the second science paper. And in 2010, PacBio then became a public company. And we did have some early successes. We had some high-profile papers looking at microbial outbreak strains, some short fragments in cancer, phasing mutations. The methylation detection was demonstrated early on. But there were 3 fairly serious limitations. The reads were not long enough, the reads were not accurate enough, and they were just not enough of the reads in terms of the throughput, which made it far too expensive. And so for the next decade, really, we then worked on overcoming these 3 limitations. The first one, the read length, that the reads were not long enough, was largely solved with an R&D with a nucleotide engineering. Early on, I had recognized that this particular configuration, where the label is on the phosphate end of the molecule, would be preferred in the method. And by 2011, we had generated these molecules and had done some optimizations. But the problem was that the label, the fluorophore, was very close to the active site of the polymerase. And so now when you shine light on this with a laser, the polymerase essentially got electrocuted very quickly and stopped sequencing. In something that I think is a somewhat underappreciated marvel of organic synthetic chemistry and biochemistry, by 2015, the nucleotides looked something like this. So we were able to not only physically move the label away from the polymerase with all these scaffolds and encapsulate, we were also able to generate some scaffolds around the label. So if something did get electrocuted, it wasn't the polymerase, but it was a part of the fluorophore and the nucleotide analog itself. Then I think this is very well known. The accuracy was solved with the introduction of HiFi. Before 2019, the world of sequencing was really divided into 2, as you know, the short accurate reads, which we still have from Illumina, and then long, noisy or error-prone reads. And by all accounts, Shaquille O'Neal was, shall we say, an error-prone long-distance shooter. And then, of course, with the introduction of HiFi reads, that paradigm was completely changed, where as we were able to increase the fragment lengths, the insert lengths, we could keep the accuracy high and generate reads that were just as accurate as the short reads. And that's because we uniquely sequenced the same base of the same molecule multiple times, both strands. And let's see whether Steph Curry is still going to make this one. Of course he will. That's HiFi. So some people immediately realized this. Is Gene Myers, of course, one of the principal architects of the first human genome, of the Human Genome Project. When we sent him some data in 2018, he said, "Thank you, Martin Pippel, his student, and I are all lit up about it." And then, of course, in 2019, with this landmark paper, we, with the community, were able to demonstrate that this is really a new way to sequence DNA. The most remarkable evolution in PacBio's history to me is the increase in multiplex. You see, when I started at Cornell in 2000, I was looking at 1 ZOMA waveguide at a time. And if you missed it, there it is. And my daily throughput was about 10 to 12 ZMWs per day, because the reaction would only last about 30 seconds, and then I had to start over and regenerate everything and so forth. I also had to work in complete darkness most of the time because, as you see here, it's an open system. There's no lid on it and so forth. By 2005, we were able to, this was my first AGBT conference, and we proudly presented to the maybe 150 or so attendees, with a very different meeting back then, that we had a multiplex factor of 19 by 19, so we were looking at 361 ZMWs. We demonstrated smart sequencing on an array with these machines of 3,000 ZMWs monitored simultaneously. And the only other thing I'll say about this, my life quality and my vitamin D levels increased dramatically with this instrument, because for the first time in over a decade, I was able to work with the room lights left on. And then a lot of engineering went into the first commercial system, the PacBio RS. So you can see up there, we were putting each ZMW onto these little cones, nanofabricated cones, to increase the optical efficiency. And so we had what at the time felt like a remarkable multiplex of 75,000. Unfortunately, this first commercial system also represented a dead end, or something that you couldn't actually increase further. So we had just had our first commercial system, and we were hitting a roadblock. So you see, to increase even further, we filled all the screen with ZMW, so I'm going to shrink this quite a bit to enable more multiplex. And so we basically had to completely re-engineer the system, with the SQL system coming in from the side, with the illumination, with waveguides, and then having the sensor as part of the chip, and so forth, and really being able now to increase it to 1 million. Now that architecture was scalable, so soon thereafter in 2019, we're able to increase it further to 8 million, which was the Sequel 2 instrument, which was launched at the ASHG conference in 2019. So now we filled up the screen again, so I'm going to shrink it down one more time, you get the idea, and further now increasing it to the Revio system with 25 million. And as you know, there's 4 smart cells that are viewed simultaneously for an effective multiplex of 100 million reaction sites monitored simultaneously. So 100 million throughput increase over 22 years, I think is pretty remarkable. And of course now, since the introduction of Vega last year, we now have a multiple product portfolio, multiple systems that you can choose depending on your throughput needs, capacity needs, and I think that's just a tremendous and really remarkable journey. So now switching to the impact, we're going to see a remarkable 2 days of all the discoveries and insights that you have made using the technology. So I'll be brief with my remarks and just highlight 3 examples. The first one is that HiFi sequencing has fundamentally changed the notion of what it means to sequence a whole genome. Other people have commented on this. Here's Gene Myers again, who 10 years ago commented that short-read whole genome sequencing generates what he called Swiss cheese genomes. They're full of holes. Other people like Debbie Nickerson or Sir John Byrne have commented that the W in whole genome sequencing is missing. So there, oops, there goes. And in contrast, HiFi sequencing not only gives you a flavor of cheese that doesn't have any holes, but it gives you 2 wheels of cheese. And of course, representing the haplotypes, the copies that one copy from your mother and one from your father. And it is so important to detect and phase the subtle differences between the 2 alleles that is so important for understanding recessive diseases, allele-specific methylation and imprinting, allele-specific splicing, and so forth. And so the ability to no longer collapse those 2 haplotypes, but to separate them into 2. Or 12, for that matter. Sugarcane is probably the most complex genome that was ever assembled, and HiFi made that possible. Sugarcane is dodecaploid, so it has 12 copies of each chromosome, but not all the time. I guess if you're sugarcane and have that much, you know, every once in a while you miss a copy and no one is the wiser. This was finally solved and assembled in an international team led by Hudson Alpha Institute for Biotechnology last year, and it's just a tremendous advance in the area of agrigenomics and plant animal sciences. 10 years ago, I talked to many plant animal scientists, and they had largely given up trying to assemble their genomes with short reads. And so there's been a tremendous renaissance. Every crop, every livestock species now has a PacBio-based reference genome. Increasingly, we're seeing pan-genomes to really capture the full genetic diversity of a species or a clade. And all the major biodiversity initiatives are using PacBio as the core technology. The second one in the area of RNA research is that HiFi sequencing demonstrates more and more that the term gene expression is really a meaningless term. Why is that? Well, over 90% of human genes are alternatively spliced, and on average, there are 7 unique transcripts per human protein coding gene. These 7 transcripts, these 7 isoforms also don't have slightly different variable functions, but it's kind of the same. No, they behave as if they're encoded by distinct genes, and there are many examples now where 2 isoforms from the same gene have the opposite biological function. BCLX in the cancer space has been well known. It was in all the textbooks, and PacBio sequencing has contributed many more examples where isoform 2 increases a certain biological function, and isoform 1 increases certain biological function. Isoform 2 of the same gene decreases that function. So if you now measure with RNA-seq just the tags and measure gene expression, you haven't learned anything because you don't know which isoform is affected, which biological function is affected. This is something that I think is not as well understood and recognized by the community, so we could all use your help to raise more awareness of this fact. But it is happening that it is, of course, gene isoform expression that is the biological driver of biology and disease. And I was very happy to see Dr. Colette Felton a few months ago at the ACMG meeting, that's the big clinical meeting, commenting that short-read RNA-seq is like the famous fable of the blind Buddhist monks. In this case, I chose a picture of a blind scientist touching different parts of an elephant and trying to reconstruct. And she said, with PacBio, you just see the whole elephant. And Dr. Felton is here at this meeting. She's giving a presentation, and I'm very much looking forward to learning more about her research then as well. Lastly, this is a project that I was involved in most recently, is to synchronize multi-omics. Traditionally, you need a lot of different assays, you have a lot of different files that you have to put together. And about 2 years ago, I called Andrew Sturgakis, who is the inventor of the FiberSeq method, and that's for chromatin, the chromatin architecture, determinative HiFi reads, and asked him whether he'd be interested in collaborating on trying to put all those omes into a single SMRT cell. At the time, I wanted to call it the PacBiome. That did not survive, but that's a good thing. That's why I'm not in marketing. But it worked really well. And a few months ago, the paper came out in Nature Genetics demonstrating that it is possible to get 4 high-quality omes from a single Revio run: the genome, the methylation, the chromatin architecture, and the full-length isoforms, the transcriptome. This is figure 1 of the paper, and when we were working and generating this figure and I saw this for the first time, I thought, yes, this is what I've been dreaming about for the last 25 years, because that is pretty much everything there is to know about the nucleic acid state of a sample. You have the 2 haplotypes, one in red on the top, one in blue on the bottom. Each of them have 4 tracks, the genetic variants of all types, the methylation, the chromatin, and the full-length isoforms. And so we then used this the new multi-omics and applied it to a rare disease patient. Oh, yes. So since the paper came out, Andrew has already demonstrated that in addition, the FiberSeq data can be used to get information about the 3-dimensional structure and the chromosome associations of the sample. Okay, now to the rare disease case. Oh, okay. So he also demonstrated that it is possible to get a full genome-wide view of all the occupancies of the different transcription factors on the DNA as they exist in the cell. But now, as promised, the rare disease case. Okay. So Scott Geip at the plant-animal meeting described his efforts to incorporate Kinect's 16S information by taking a sample and at the same time get high-quality information from the gut microbiome. So you see that very quickly after the first paper, you all are already innovating and adding even more omes to enhance the capabilities even more. So now, yes. So we applied the original 4-ohm example to a 9-month-old girl, part of the undiagnosed disease network, and there were a lot of health problems, as you can see on the left with this little girl. It was undiagnosed, and so really showing in this table that you need all these high-quality views of the genome, the transcriptome, the methylation, the chromatin, to fully understand the molecular reasons for the different phenotypes. And to me, this was a great example of what it means to enable the promise of genomics to better human health, to really get a complete understanding of what is going on. Biology is very complex, and you need better tools than short-read sequencing to illuminate them. So with that, I'd like to close, but not before thanking all the colleagues at PacBio over the last 25 years, and there have been over 2,000 of them, for their hard work, companionship, and incredible journey. My talk was relatively technical, so I do want to stress on the right that it's not only the scientists, it's all these different departments that have to come together, that have to work together to create a company that can bring these technologies to you. You see on the very bottom right, it says partners, and so I do want to stress the importance of all the partners that we need in our ecosystem of the PacBio compatible, and specifically the sponsors for this meeting. We have 13 of them. They all have tables out there past these doors, so please go check out the table, talk to them to make the full workflow from the sample all the way to the data analysis the most efficient that it can be. And lastly, thank you. We would not be here without all of you. We thank you for your business, for choosing us as a partner in your research. And as a thank you slide, I have here a graph that shows the accumulation of PacBio data since the beginning of HiFi in 2019. You see there's a very steep increase in the curve. Last month, you all exceeded 130 petabases. That's 130 million billion bases of PacBio data that was acquired. If you have contributed to this graph, thank you very much, and we are deeply indebted to your partnership. The other thing that I want to say that I find amazing, that over half of all the PacBio data that has ever been assembled and generated just happened since the beginning of last year. So with that, I'd like to invite you to celebrate with us, and let's have some cake. I will in a moment ask Christian to come up to the stage and cut the first ceremonial piece. We will then move the cake down to the reception area, which starts right after this event, and you can all have a piece and join us in the celebration. Once Christian is finished with the ceremonial first cutting, we would then like to ask all of you to move to the center of the room so that we can take a group photo and capture this moment in the journey, in the history of our company. There will be a photographer here, and so that we can capture this moment. So with that, I'd like to thank you very much. And Christian, if you could come up and do the honors.
We need a circulomics kit. Does anyone got a circulomics kit in the house?
There you go. Yeah, beautiful.
There we go. Oh,
nice.
Confetti cake. Yeah, baby.